Good afternoon. Yeah. So, Cyclin-Dependent Kinase-9, together with the cyclin T and some transcription factor, form a complex that is named positive transcription elongation factor B. That this complex phosphorylates serine 2 within the carboxy terminal domain of the RNA polymerase II, allowing the elongation activity of transcription. This complex is present also in an inactive form, and in this case, with the lower kinase activity in this complex, there are, in addition, EXIM in its isoform 1 and 2, the 7SK small nuclear RNA, and the LARP7 LA-related protein, LARP7. And this molecule enabled the repression of a CDK9. And many compounds have proved to inhibit cyclin-dependent kinase-9 by competing for the active site. But one of the most specific is flavopiridol. So CDK9 has been shown to be increased in cardiomyocyte hypertrophy, and that is a fundamental response of the stressed or injured heart. Knockout mice for EXIM, that is a CDK9 repressor, cause a severe cardiomyocyte hypertrophy. But we know that many of the pathways associated with the cell division are also known to be activated in this hypertrophic response, leading to the hypothesis that mammalian cardiomyocyte hypertrophy might be the result of a block in choreo and cytokinesis. So what could be the role of CDK9 in cardiomyocyte proliferation? Some recent papers have shown that by silencing CDK inhibitor, mice cardiomyocyte re-enter the cell cycle. And among them, CDK9 act as a cofactor for activation of transcription by myocyte enhancer factor 2, and also CDK9 form a complex with GATA4 in cardiomyocyte, and GATA4 is a well-known driver of cardiomyocyte proliferation in mammals and also in zebrafish. Zebrafish that unlike mammalians, retains an impressive ability to repair the heart following injury. Therefore, I hypothesize that CDK9 is a master regulator of cardiomyocyte proliferation in the zebrafish during the normal development and affects the recovery following a cardiac injury. So, firstly, I characterized the response of the response and the recovery ability of the zebrafish embryonic heart to the injury. I used a laser to induce the localized and controlled injury to the ventricle of the CMRC2 GFP zebrafish embryo heart. So ejection fraction as a cardiac function was assessed by the diastolic-systolic area subtraction method. For cardiomyocyte analysis, hearts were surgically isolated and stained with DAPI for total cardiomyocyte analysis, and with antiphosphohistone H3 for mitotic cardiomyocyte. Images were collected under confocal microscope and analyzed using IMAGE-J software. So this is the zebrafish at zebrafish embryo at 72 hours post-fertilization, and the major organs are already formed. The zebrafish embryonic heart contains only a few hundred cardiomyocytes, which can be readily counted and makes this a potentially accurate and reproducible model of a cardiomyocyte proliferation. The zebrafish heart is two-chambered, so a single atrium, and the ventricle seen easily in this video. And this is the zebrafish line, transgenic line I have used in my experiment. So I used the embryos aged from 0 up to 120 hours post-fertilization, and at 72 hours post-fertilization, a single laser beam was delivered into the ventricle, and the measurements were assessed before laser and then 2, 24, and 48 hours post-laser injury. In this short video clip, you can see the laser technique. So this is the ventricle, this is the atrium, this is the bulbus arteriosus, and a single laser pulse is delivered into the ventricle, and significant injury is confirmed by arrest of the ventricle while atrium continues to beat. So for the cardiac function, ejection fraction was assessed for each individual embryos at each of these 4 time points. In fact, these black and white videos captured under fluorescent light showed exactly the same ventricle at a different time point. And overall, following laser injury, you can see that the cardiac function showed a dramatic decrease at 2 hours post-laser. At 24 and 48 hours post-laser, injured embryos show values of ejection fraction comparable to control. Here in this graph, gray bars show the increase in cardiomyocyte number during normal development, and in blue, following laser injury. So laser injury produced a 15% of reduction in total ventricle cardiomyocyte number, while at 24 and 48 hours post-laser injury, the cardiomyocyte number was approximately the same in both damaged and control groups. And here above, there are some imaging showing some DAPI stained ventricle on a GFP background, and on this second image, the typical loss of GFP signal by the laser injury. So in the following series of experiments, I used the same protocol as before, but in this case, zebrafish embryos were exposed to a low dose of flavopiridol at a concentration of 3 micromolar from 24 hours up to 120 hours post-fertilization. So again, this is the curve of ejection fraction during normal development and following laser injury. So you see a decrease in cardiac function following laser injury and recovery at 24-48 hours post-laser. Laser injury in embryo exposed to flavopiridol also caused a significant reduction in ejection fraction, but in this case, no recovery was observed. A similar trend was observed for cardiomyocyte number. So in embryos not exposed to the drug, there was a short reduction of a number following laser injury, and the return to normal at 24-48 hours post-laser. While the embryos exposed to flavopiridol, the cardiomyocyte number was reduced at baseline and remained low up to 48 hours post-laser. These images show the normal pattern of a mitotic figure during normal development, shown as a pink-red spot by phosphohistone 3H3 staining, and these one following flavopiridol, these are reduced. So this is summarized in this graph below with the normal changes between normal development and the following exposure to flavopiridol, that this mitotic figure are significantly reduced at all time points. So I have demonstrated here a novel model of laser-induced cardiac injury in the zebrafish embryonic heart, and the remarkable ability of the embryonic heart to recover cardiac function within 24 hours. This could be due to a high background of cardiomyocyte rate in the embryonic heart. I have shown that CDK9 inhibition by using flavopiridol significantly impair functional recovery following laser injury and also inhibit cardiomyocyte proliferation. Inhibition of cardiomyocyte proliferation by flavopiridol could partially explain the failure to recovery of cardiac function following laser injury, but other effects of CDK9 inhibition on cardiomyocyte function are not completely excluded. And this data suggests that targeting CDK9 is a promising strategy to modify injury and recovery in the heart. The result I have shown here, achieved by pharmacological inhibition of CDK9, need now to be confirmed by a genetic inhibition by CDK9 morpholino. And I also want to take out one of the breaks that maintain the CDK9 in a low active state. And I'm using now LARP7 morpholino to inhibit LARP7. And this is the last slide. So I want to show as the last slide just this video. This is an improvement that we have recently done on our model of laser injury. So when the embryo is placed on the slide, it naturally plays on a side. And the atrium and the ventricle are often overlapping. Atrium and ventricle are often overlapping, particularly during the ventricle systole, that make difficult injury of the ventricle without damaging the atrium, countering then that embryo as a valuable experimental sample. Now, and also the fast systole-diastole cycle in the zebrafish heart, that in this case is a threefold less, affect the accuracy in delivering, in delivering the laser beam in a high specific cardiac area. So to overcome these problems, we are using now a laser ablator in combination with a synchronization system. So in this case, we can set up the software so that we can fire the ventricle at the end diastole phase. And moreover, by hitting the ventricle in the diastole, we can select very few cardiomyocytes because they are relaxed, and so very few cardiomyocytes can be ablated. And this is important if I want to study few cardiomyocytes, as someone has already shown in this very beautiful paper with the multicolor clonal analysis, and they have started to define the contribution of individual cardiomyocytes on the zebrafish cardiac development. So I would like to thank my colleagues that help for their help, and the British Heart Foundation and the University of Edinburgh for funding my work. Thank you very much.